When you think of sexual selection, you might think of deer budding antlers or a peacock displaying its plumage. These both represent pre-copulatory forms of sexual selection, and while important, don't fully describe the origin of a progeny's genetic makeup. This is because in some species, there are also post-copulatory factors that play a role. In Drosophila melanogaster, gene-encoding sperm components and seminal fluid proteins influence refractoriness, egg laying, and sperm storage in females. However, these all result from the competitive success of one male's sperm against their rivals. Do females have any role in sperm competition? Short answer, yes. Proposed by Eberhard in 1996, Cryptic female choice influences which male's sperm fertilizes a female's eggs. Why is this an evolutionary advantage? Well, unlike human beings, female fruit flies don't usually mate with one male, or even two. They mate with several and store sperm, increasing competition for egg fertilization. Longer sperm are better at displacing competitors, but because creating them requires a greater energy investment, only large and healthy males are able to consistently invest in their creation. Therefore, the fact that females have influence over sperm ejection, storage, and displacement is hugely impactful on the resulting offspring. So to investigate this, a 2013 study by Chen and others analyzed these organisms and identified single nucleotide polymorphisms, or SNPs, within females via a genome-wide association study. These SNPs caused variation in the relative number of offspring which were derived from the first male. While the role of these genes are unknown for sperm competition, post-mating responses have already been linked to activity in DH44, TDC2, and PPK neurons. Because 15 of the 33 genes found are linked to the nervous system, or neural functions, it is possible that the female nervous system directly plays a role in modulating sperm competition. This brings us to the study's main objective. What Chen and her colleagues aimed to do was identify which genes, originally proposed by Chow's team, affected sperm competition, and if it did so through the female nervous system. For their materials and methods, candidate genes were knocked down in females by RNA interference and the UAS-Gal4 system. Driver lines were ubiquitous or nervous system specific, and UAS RNA lines were from ATTP and W1118 backgrounds for each candidate gene. How does this process of gene knockdown work? Well, first of all, UAS stands for Upstream Activation Sequence. Gal4 is a transcriptional activator that binds to UAS sequences in DNA, inducing gene expression. For gene expression to occur, a Gal4 driver fly, which expresses Gal4 in a specific tissue, is crossed with a fly that has a gene under control of a UAS, resulting in their progeny expressing the gene of interest. However, if we want to knock down genes, we couple the UAS Gal4 system with RNAi. In this case, the UAS fly has an inverted gene sequence prior to the actual gene of interest. When transcription occurs, a double-stranded RNA hairpin is created, resulting in degradation of a tissue or cell-specific mRNA, and thus knockdown is achieved. In Chen's sperm competition experiments, both control and knockdown females were kept in individual vials and were made to CMBW, cinnabar brown males, followed by BWD, brown dominant males. After the matings, females were moved to empty vials and the number of offspring from each male were tallied for analysis. Eye color was used in categorization of offspring, as female progeny of CMBW males have red eyes, while female progeny of BWD males have brown eyes. Because the eye color gene is located on the X chromosome, the males were not used to assess paternity. In terms of results, the parameters they were looking at were remating rate, the chance a female will go and mate with the second male after mating with the first male, fertility, and P1, which is the number of female offspring sired by the first male compared to the total number of female offspring sired overall. Because the experiment was focused on sperm competition outcomes, only doubly mated knockdown and control females were considered into their analysis. What the researchers found from knocking down 27 genes is that 6 resulted in a reduced remating rate. Only TDC2 and PPK neuron-specific knockdown of the HID gene led to an increase in the remating rate. In terms of fertility rate, knocking down genes in the drivers resulted in a lower rate for every gene, except ZASP66, which is ubiquitously knocked down, and HID, which is PPK specifically knocked down. The last figure shows the effects of gene knockdown on sperm competition outcome, specifically on P1 over the course of 8 days. In the shown figure, each graph represents the knockdown of a gene that affected sperm competition outcome using a driver and RNAi. Each dot represents a trial. Orange dots represent the knockdown groups, and gray dots represent the control groups, from which an ATTP or W1118 background strain was crossed with an appropriate driver line, resulting in lethal knockdown. In each of the figure's plots, mean error bars are displayed. The researchers determined the differences between the error bars, as well as the distribution of P1 between knockdown and control females, to determine p-values, a measure of statistical significance. What they were trying to do is find out what proportion of offspring are from the first male, and in every case, with the exception of knockdown in HID and MSP300, the P1 dropped. So where does this bring us? What does this information mean? What the researchers accomplished through this study is showing that the female nervous system has a profound effect on sperm competition through the knockdown of genes within neuronal drivers. They also demonstrated that knocking down certain genes can increase or decrease female remating rate as well as fertility. In terms of difficulties the researchers faced, RNA lines for two genes, SK and CG33298, gave insufficient RNA knockdown for testing and thus were unable to be assessed for their role in sperm competition. Some knockdowns also have the potential to affect female receptivity to remating, or could have detrimental effects on overall female health and development. This means results could be either direct or indirect consequences of gene knockdown. In terms of difficulties a reader might encounter, a lot of terms and processes were assumed to be prior knowledge, such as the UAS-Gal4 system, RNAi, driver lines, and allele naming conventions. It is highly suggested that a reader inexperienced in genetics review these terms for a clearer understanding of the paper. What are the next steps to take from this study? The researchers ran into some problems with analyzing their genes of interest, because the Vienna Drosophila Research Center didn't have suitable UAS RNAi lines for 5 of the genes. Another 2 genes were knocked down, but didn't give any detectable signal. Therefore, future studies that could be done should gather data for the genes that were unable to be investigated. Another possible study that could be looked into is the mechanism and effect of the ZASP66 gene, which when knocked down, increased fertility. If there is a human gene that demonstrates a similar effect when knocked down, it might be possible to develop a gene therapy treatment that could potentially increase fertility rates in human females. In conclusion, it is clear that this study helped illuminate the role of the female nervous system's contribution relating to sperm competition. As is the case with all science, more research is needed for more definitive answers.